Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. This is a very special episode of Careers in Discovery because for the first time ever, we have a brand new host. This week, taking the chair is Emma Hobbs, one of our people and culture partners here at Singular, and she's with Torleif Gjølberg, CSO and co-founder of Authera, a company innovating in antibodies and albumin-based therapeutics. Join Torleif and Emma as they talk about his fascinating career so far.
So, hi and welcome. This week, I'm delighted to be joined by Torleif Gjølberg, the CSO and co-founder of Authera. He's joining us this week on Careers in Discovery. Welcome, Torleif.
Thank you. Happy to be here.
And thank you for joining us. I suppose the best place to start is by talking a little bit about Authera and what the business and the mission is up to at the moment.
Yeah, so Authera is a 2-year-old company. We are still fully owned by the co-founders and the scientists that founded the company and the people that work here. And what we are doing is all centered around antibody-based therapeutics, antibody and albumin-based therapeutics. So it's all about proteins and biologics. And the core expertise of Authera is related to the therapeutic performance of such molecules, specifically how it's dictated by their interactions with an antibody and albumin receptor, which is called the neonatal Fc receptor. Okay. So that's abbreviated FcRn for short. And it's quite central in the therapeutic performance of, I would say, the vast majority of all antibody-based therapeutics that has entered the market. So basically what this receptor does is that it prolongs the circulatory half-life of both IgG-based antibodies and albumin. So these two evolutionary non-related proteins have evolved to interact with the same receptor, which is quite fascinating. And we experience that sometimes people are very aware of this and sometimes people are not so aware of this. But in the case that you have had therapeutic success with an IgG-based molecules, it's most likely because it has an interaction with FcRn. So that's a bit of the science that Authera is founded on. And the way we leverage that is that we have a lot of knowledge regarding the biology of this receptor coming out from a few decades of academic research. Mm-hmm. So Authera spins out from an academic research group at the University of Oslo in Norway and Oslo University Hospital. The research group is headed by Professor Jan Terje Andersen, which is, I would say, a renowned expert and key opinion leader within the FcRn field. Okay. And previously, the research group was led by Professor now Emerita Inger Sandli, who has also co-founded a few other biotech companies in the past. So spinning out from that research environment, we are taking with us a lot of scientific know-how and specifically assays that tackle various aspects of FcRn biology. And we have integrated those methods into one methodology platform that can tackle everything from biochemical interaction studies to tailored cellular studies, which is one of the big strengths in Authera, to also suitable in vivo studies, with the overall aim being to try to predict pharmacokinetics and biodistribution of antibodies and albumin at very early stage in preclinical development. So by using those methods, we think that you can de-risk your clinical pipeline. Yeah. And you can learn information that you would like to have that sometimes you get late, but it will be better to get it early so that you can narrow down the scope of how many molecules you need to proceed through different testing. So that was a long way around to getting to what we actually do at Authera, which is that we have 2 business arms. One is that we leverage this method platform in collaboration with industry. So we open this methodology platform to industrial entities that are developing antibodies or albumin-based molecules. Yep. And we experience that that's a nice way of working. I mean, we get to know them, they get to know us, and then we funnel the income that we get from that part into our therapeutic development. Okay. So we are doing both. So one is more collaborative, the other one is therapeutic development. And in therapeutic development parts, we are developing an antibody-based molecule that is intended to treat exudative eye diseases.
Okay. Very exciting. And for such a young company to be having revenue streams already, that's great, great success.
Yeah, we're actually quite proud of that. We usually say that it's quite unique, which is a statement
Absolutely.
I'm not sure if I really have the data to back up, but usually biotech companies they are funded on an idea, and then you get some seed money in, which is great in many ways. I mean, you get a lot of expertise from investors and that sense. But in our case, when we spun out the company, it's headed by myself and a friend and colleague called Simone Mester, and my co-founder and CEO. It's also co-founded by Authera and Inge Sandli, the professors that I mentioned. So we are the 4 co-founders. And a big thing for us spinning out from academia was that we really wanted to learn like the ropes of how to not only run a business, but also to establish it, the economics of everything, and do it all. And we didn't really start. We started with a lot of ideas and a lot of know-how, but without any hard IP, so to say. So that is being generated now. And in that sense, we felt that if we can build value within the company with revenue, maintain control for a few years, then we will have a very different concept to pitch to investors by the time we do need them, because we will need them for sure, especially for the therapeutic development part. I mean, there's no way we can finance that ourselves, of course, but so we're very happy about that.
Sounds like an excellent business model. I'm really keen to hear more about your background, particularly the reasons of why you chose a career in science.
Yeah, so my background comes from molecular biology and biochemistry. Initially, during my studies, I did a bachelor's in molecular biology, and it was all very theoretical. So I started university now 12 years ago, and for sure, I mean, the educational run that I did has changed a bit since then. But when I did it, it was a lot of theory, sitting in the study hall and, going over the details of cellular signaling mechanisms and everything. I found that very interesting, but I also found it a bit hard to see, where could you go with it in a way. Most of the role models that were around were very good professors, PhD students, people that were sort of doing research, academic research. In Norway, it wasn't really at that time a very robust, I would say, biotech-centered pharmaceutical scene. It's still very much growing, but it's not like we have a lot of R&D-driven biotech like you would have in the UK, for instance, when you have big like AstraZeneca campuses, or there's a lot of good big pharmaceutical companies in the UK. I mean, they are here too, but most of them are not doing active research and development here. They are doing more sales and clinical stuff. So in my bachelor's, I started to feel a little bit like, I want to see if I can apply this to something. So when I was looking for a master's, I ended up at a more patient-centric research institution, because that felt like it was stepping more into, okay, I know all this stuff now about cells and everything. Maybe I can apply it to help someone.
Yeah.
So I ended up making a change. That was all what it was all about, right? So I did my master's then at Institute of Cancer Research here. In a very nice research group at the Department of Radiation Biology.
Okay.
So then it was all about cervical cancer and prognostics, which for me felt a bit more, as I said, translational. I was allowed to come into the room with patients that were actually, the people that you were doing sort of research to help, which was a bit of an eye-opener to me.
Yeah.
I really liked that. But I still felt like, even if I stay within research, the impact that I would be making would be very diffuse in a way, vague, probably years down the line. I mean, you publish something and you hope that people will read it, or you have an idea and you take it to the tech transfer office and you hope that it comes something out of it. So I started to feel like I wanted to do something here and now. I'm a bit impatient.
That's not a bad quality.
I suppose as an entrepreneur, it's not that bad quality, So some friends talked me into joining an innovation competition as a student, which is called IGEM, stands for Internationally Genetically Engineered Machine. which is a very cross-disciplinary international thing. So the idea there is that you get students together from different backgrounds, and you're supposed to solve a problem by using synthetic biology. And the problem is yours to define. I mean, and the project is yours to define. So I thought that was quite cool. We pulled together some students from molecular biology, some pharmacists, some informaticians, some physicists, and we built a project to speed up diagnostics of antibiotic-resistant bacteria, which was very much on the side from everything I was doing in my master's. So I thought it was quite cool, and we succeeded in the project to build a functional prototype for a device that could detect a certain type of broad-spectrum resistant bacteria before using any antibiotics. And then we were allowed as students to travel to Boston from Oslo and pitch this to a panel and be part of like this huge jamboree thing, which was a very cool and I would say pivotal experience back then. You know, I was, I don't know, 21, 22. So to be allowed to come to Boston, sort of the mecca for biotech and pharma, I suppose.
That's an exciting experience.
Yeah, that was quite neat. And since the prototype was functional, it was possible to make it into product in theory. So when we came back from there, actually the tech transfer office here at the University of Oslo, they reached out to us and said, well, I mean, you have this nice gold medal from this competition, you have this functional prototype, would you like the chance to try to start a company around it? And of course, if someone asks you to start a company, my gut was like, hey, you can't say no. I mean, that's a cool experience to have. So we said yes, and I served as the CEO and chairman of the board of that company.
Yeah.
So it was a very small medtech-based startup. We were sort of just, trying to do something. We didn't really know what we were doing. To be honest, we were just...
You were so young, yeah.
We were very young, and we were still doing our masters. So this was all like in parallel with our studies on the side. So what we did in the company was that we wrote a business plan, and we started submitting that business plan to a lot of pitching competitions just to get feedback from people who actually knew business, because we were acutely aware that, we know some science, we are still like below graduate level, and we definitely don't know the business. So we wrote a business plan, we entered some competitions, and we were able to win some award money that sustained the company for some time. And I felt for myself that I learned quite a bit about being in a team, shareholder structure, company structures, writing a business plan, pitching it. I was the one doing the pitching. So I got to face some investors, everything from, it was mainly angel investors back then. So some consortiums where 20 angel investors come together, they all have a little bit of money, and you pull it together to create one big award for the company that wins. Some competitions we went well, some competitions went fine, not so well, and that's perfectly fine. That was part of the learning.
Yeah, absolutely.
And we also were able to secure a spot in some more business education things that the company could be part of. So I felt, for me, I was starting to build more of both an interest and a knowledge within the business side of things. And then I completed my master's, and it was sort of becoming clear that the company was not really going to be a big success unless someone, took the leap and quit their jobs, started working on it full time, all that stuff.
Yeah.
And due to the history of how the company came to be, starting with a public disclosure of a prototype, it wasn't really the best case for anything. So, and also actually a feedback that I got from a lot of the investors was, you are very young. And usually for companies such as this, we bet that the founders have the scientific know-how and we come in with the business. But then typically that person is a bit older and they have a PhD. So have you been considering doing a PhD? And if you would do a PhD, how would that look for the company? And I met that feedback time and time again, that a PhD would really solidify our trust in you, which was fair.
Is that something you agree with?
I'm not sure. I think it depends a bit on what you're doing. For biologics and biotech, I mean, if you're developing an actual drug that is going into someone, I think doing a PhD matures you as a scientist in a lot of ways. There are very few positions or times in a person's life when the sole thing you're supposed to do is to be learning, and a PhD student is one of those positions.
Yeah.
So I think a PhD can be a lot of different things for different people, depending on where you do it, how you tackle it, what your project is, all those things. So I'm not sure if I fully support the idea that a PhD automatically gives you credit or weight in the room.
Yeah,
But
it shouldn't tick the box.
yeah,
Yeah.
I'm not sure if it should be a tick box. Yeah, that's a good way of saying it. I think you can get a lot of the same know-how if you are at a good company or a good institution and you're allowed to grow as a scientist along the way. But for sure, for me, doing a PhD was a good thing. So I started looking around for PhDs, but still being impatient, I wasn't really in the mood for a really basic research project that was far away from both the patient and the innovation that I'd sort of gotten a taste for.
Yeah.
So I was looking for a PhD in an environment that would allow me to work in an innovation-centered way,
Yeah.
to put it like that.
Something really get your teeth into and make a difference again. Yeah.
Yeah, something like that. So, and then there was an opening at Jan Terje Andersen's lab. It was actually a bit funny because I think the job ads it was very vague, reflecting also that the project was sort of to be formed a bit by the candidates coming into it. But I was talking to Simone, my co-founder. She was doing her master's in that research group, and she opened my eyes a bit as to what they were actually doing in that research group. So like, okay, maybe I'll apply. Maybe they want me. We'll see. But I was very much like, okay, I was also fine with being where I was. And the plan was to do a PhD in the research group where I did my master's, because it was a very good research group, very good people, and I really liked the patient translational part of it.
Yeah.
But well, I mean, the position was there, so why not apply? So I applied, and I sat down in the interview, and I told them, well, I could envision myself doing a PhD with you, but not because I want to be a researcher. It would be because I want to start a company together with you at some time, either during my PhD or after my PhD. I knew that Jan Terje was doing a lot of really cool, innovative stuff with antibodies. I didn't know if he wanted to start a company, but I thought that maybe he would be interested if the right people
and you're
came
persuasive.
Well, I tried to be, at least. Later, he has told me that he remembers very well this young person without much experience sitting down and saying that I know who you are and I want to start a company. But yeah, maybe it was a bit bold back then, but at least it got me the job. So that was good.
Yeah.
So I started my PhD, and it was a very close collaboration between the basic research environment from Jan Terje, and that research environment is one of the most innovative in Norway. I'm very proud to be a part of that research environment. Jan Terje and Inger are among the professors in Norway with the most registered innovations ever. Okay. So they have a lot of ideas, and they have a lot of experience when it comes to collaborating with both the university and industry on translating those ideas into something more than just a research project. Yeah.
And it's so important to bridge that gap between academia and industry, isn't it?
Yeah, for sure. I was really fascinated by how that works. Coming from a state-funded research environment, how do you actually translate that into commercial value and a product that can help the patients in the end? So it was close collaboration with that basic research environment and the Department of Ophthalmology at the hospital. So in that sense, it was, again, a very patient-centric thing. Yeah. And my PhD position came about after the research environment at those two, so the clinicians at the Department of Ophthalmology and the researchers at Jan Terje's lab, they came together to solve a very concrete problem that the patients, or actually the clinicians, were having, which was related to how you prepare syringes that contain antibodies for injection directly into your eyes. So it's called intravitreal injection, and you do this with antibodies targeting certain things to treat certain eye diseases. Yeah. So there's certain very prevalent eye diseases, and the label dictated I think it still does, actually. The label dictates that the doctor is supposed to do the syringe withdrawal from the drug vial in the room with the patient, so that you know that the vial is not compromised before then. Yeah. obviously, because the needle is going directly into your eye, you want to be as sure as possible that there's no contaminations or anything.
Yeah.
Because the moment that you withdraw the first syringe, then the vial is compromised because then the content is exposed to the air around you. So then the rest of the vial has to go into the trash.
Wow.
So there's two things about that that's not so nice, actually. Three. One is that it takes the clinician some time to withdraw the syringe, which takes focus and time away from the patients. The other is that the patient room is not sterile. So even though, I mean, there's a minute chance, I mean, there is a chance that something could be circulating or whatever that could create a suboptimal sterile environment.
Yeah.
And then the third part, obviously, and the big one, is that each vial, for different reasons, contains enough drug for 2 to 3 injections.
And they're only using it once?
They were only using it once, so they were throwing it out, the rest. That's what the label dictates out of safety reasons, which is, that's how it's approved. I won't say anything more about that, but due to this, and these drugs are very costly, as I'm sure you're aware. Biologics in general are very costly. So one vial could be priced at £1,000, 10,000 Norwegian crowns. So it was a lot of money going literally into the trash.
I don't think you need a PhD to understand that that's not logical, that the math won't add up.
It's not really logical, and it seems like such an easy thing to tackle, right? But the thing is that if you want to do this differently, then you would have to withdraw the syringes before you come into the room with the patient, so you can do it in a sterile chamber, right? And then obviously there will be a lag time between this compounding of the syringe and injection into the patient's eye. So what you then need to prove is that that compounding period on the syringe withdrawal doesn't affect the biologic that is going into the eye. So what they did was to design a compounding procedure. Let's say that you take the drug vial, you prepare as many syringes as you can, and then you take those syringes, you store them for up to 7 days. And then you take them to the people who know biologics, antibodies in this case, and you ask them, can you prove that the ones that have been stored for 7 days are as effective and safe as the one that have been stored for no days?
Immediately,
Immediately, right?
Yeah.
And they were able to prove that that was the case, which saved the hospital a lot of money, which again, a very long way to explain how my PhD came to be, because out of all those money that the hospital were saving, they were saying that, okay, this was a very quick fix to a very big problem. Maybe there is more research to be done in this aspect. So let's hire a PhD and see if we can do something together here. And that PhD was me. So I got to work very closely with the eye doctors and learn so much about eye medicine and antibodies. I thought it was really cool. And I really appreciated that my supervisors, they placed a lot of trust in me in the case that they really didn't place any boundaries on what I was allowed to do or not. So they just, like some projects, obviously we agreed on that this will be done, like more of those compounding studies for more biologics reasons. In other aspects, I was allowed to be quite free in the lab and just explore different things, which for me was a very also, again, formative experience, I think.
To me, it sounds like you very much earned the trust by solving such a huge problem.
I mean, that problem was actually solved by others before I came into the picture. So I sort of just helped with the details afterwards. So, but yeah, I think it was quite cool as such a young scientist to have that trust and to be able to explore your ideas.
Yeah.
And I think I grew on it. I grew to trust my own ideas because I was allowed to prove them and explore them. And then during my PhD, then I was also allowed by Authera to take responsibility for some industrial collaboration. One thing that for sure is something I wanted to mention is that we at Authera, and also before we started Authera, we experienced that a lot of industrial entities, they follow science quite closely, maybe more closely than at least I realized when I was still in academia. So they have the same problems as we describe in our research, predict pharmacokinetics, how to be sure that a candidate is a good idea. And they realized that in order to create a successful drug, you really need that deep biological insight into problems that you're trying to solve and how your molecules will behave in patients in the end. So they are reading publications, and we see that publications are the best PR that we could ever get. To publish something in a high impact peer reviewed journal, that's a good validation of your knowledge and your standing within something. So companies sometimes they would reach out to Jan Terje and say that, well, you have this nice publication describing this method. We have a problem that we think your method could solve. Would you be interested in helping us out with that? And Jan Terje placed some trust in me and Simone and others in the lab to help out with such projects. So as a PhD student, I was allowed to not only work closely with clinicians and again be in the room with patients and learning about how our research was doing a difference there, but also to work in close collaboration with industry and write industry level reports and to get insights into, okay, so I knew by then this is the way of academic publishing. Then you have peer review. This is the way of writing an industrial report. This is how the industry asks the questions, which there's some overlap there, but it's also different perspectives.
Yeah, absolutely. So if people are hoping to have a similar experience and you gained a lot of insight, as you said, from academia with patients and in industry in such a small space of time, what competencies do you think are important for people to be able to go out and achieve those same ambitions?
That's a good question. I think for me, it really came down to at least be very good with the science and know your stuff from the studies. That's boring but important parts answer maybe.
You need to be able to be credible, absolutely.
And you need to know that you have a certain level of confidence in that you know your stuff when you go into those rooms. And then the other part is also a very general answer, but to be a bit open-minded and unafraid maybe to try, because there's a lot of things that, opportunities usually they just don't come. You have to create them in a way, and then you have to dare to speak up in the room. At least in my experience, that's a good way of going at it.
Yeah, absolutely. When you talk about the experiences you had to take risks to be able to achieve those.
And for me, I think the most important lesson learned throughout my PhD, the student experiences and everything, is the people that you surround you with. To have good people around you, people that see opportunities that maybe you don't, and to trust each other, people that trust you and that you trust. Especially since spinning out the company, I think trust is between us as co-founders is really, really important. I'm not sure if I answered your question, but at least...
No, absolutely. Yeah. Commit to the science, know your stuff, have the confidence, don't be afraid to take risks. They're really important qualities.
And look for people that are cool not only on a professional level, but also on a personal level. In my experience, if you are in a work environment that you don't like on a personal basis, not that I have been, actually, I've been very lucky with most of the environments I've been in. It's been very good people. But I think, at least for me, that feels like a very important part of it, that you like and you look forward going into work every day. Because then if you're comfortable, at least I dare more, I want more. If I like the people I work with, if it's only about, okay, but this is only a stepping stone, it just sucks for a while now, and then I'm going on to something, my motivation at least dies in that environment. I need to be around cool projects, but also good people. I think that's really important.
And so I talk a lot about mentoring within my network. The mentors that you've had throughout your career, have they been formal where you've had that conversation about whether people would be your mentor, or have they been more informal arrangements?
Maybe more informal. As a PhD student, it is sort of both a formalized but also unspoken. Of course, your supervisors are your mentors. And I think then, we never really sat down and said, "Oh, you are my mentor." But of course, a PhD supervisor is in most ways your mentor. And I really liked the prospect of having two quite different mentors in that sense. So I had one clinician, Morten Moe, a great clinician, also placed a lot of trust in me, and then Jan Terje, more translational, but also really basic, deep-seated research. I think not very formal relationships, no. At least here in Norway, we have what we usually call a flat structure, so it's not really that hierarchical. which for me also works very well. It's not like you have this fear of authority that, I can't go into the room of my mentor or something. I think that's really nice.
Yeah.
Again, I'm not sure if I answered your question actually.
No,
but
absolutely.
yeah.
Absolutely. And surrounding yourself with people where your voice is heard. That's
that's a good way of putting it. Like people who actually listen to you and that are not afraid also to tell you that, well, I mean, that's a nice idea, but maybe you should explore this instead of that.
Yeah. Okay.
Yeah.
So we kind of touched on it already, but if you were to go back to your earlier self and give yourself one piece of advice when you were in university, what would that piece of advice be?
Maybe also don't be afraid to look outside of university for inspiration.
Yeah.
Again, this probably varies a lot between different universities and different countries, maybe where you're doing your studies. I know that it has changed now. Actually, the bachelor degree that I did doesn't really exist anymore. It has been replaced by a more, it's just been replaced by a more revamped or rethought educational program. But at least back then, it was very much this is science, 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 and then the industry is something else, intangible, like happening after commercial. And I think the interplay between those two is really important, especially if you want to do something like I'm doing, starting a company and that stuff. You can't really do one or the other. You need to master a little bit of both.
Yeah.
So maybe back then it took me some time to realize that, okay, if the research-driven biotech is not so much there, or maybe not accessible to me, maybe you create it. I didn't really think about that when I was doing my bachelor's. It came later in my master's, and I think I could have been more motivated in that aspect also during my bachelor's. I'm not sure if it would have changed much, but that's the piece of advice I would give maybe.
Yeah, absolutely. What's next? What's next for Torleif? What's next for Authera?
What's next for Authera is also what's next for me, in a way. As an entrepreneur, you sort of live and breathe your company. I think it's really, really cool to be running the company together with Simone. So for me, hopefully what's next is to grow together with Authera. For now, we have been for a long time in, let's say, very early stages of discovery or drug development for antibody-based molecule. And we are poised to make the next step in that regard. So I'm really excited to get to learn how to take things from discovery and through, candidate validation and more late stage preclinical research. I really look forward to that process, and I think we will be learning so much.
Yeah.
And of course, one big aspect of that is to formalize our interaction with specialist investors and to begin the journey on fundraising in a little bit of a different way. So far we have funded ourselves by revenue and soft funding. Soft funding is also a great experience to go through. And of course, we won't stop doing that either, but that's what's next, to sort of transform the company from mainly a collaborative and discovery company to also a drug development company, and how that will look, both with regards to what the molecules will look like and what the investors will look like and everything. That's something that I'm very eager to learn.
Yeah. Really exciting. Very much looking forward to seeing some clinical trials in the not too distant future.
Yeah, let's hope.
That's a scary prospect.
That's a scary process. So that's the goal, right? To get to a clinical trial. We really hope that we'll get there. And maybe we'll also start growing. I mean, when we started, it was only me, myself, and Simone, our CEO. And now we are 4 people working full-time in the company. So that's also something I really, really enjoy, to be more people, and the fact that we are coming together to build our own workplace, our own culture. And, we have to take conscious choices every day, every week, regarding what sort of culture are we building here. I think that's a really valuable part of creating a company or starting your own company, that you're allowed to create your own workplace in a way. That's pretty neat.
And it's really good to hear that you're conscious of that. Because there's so much focus that can go into the science and the fundraising, but sometimes cultures just develop on their own. So being conscious will enable you to have the culture that you want.
Yeah, I think that's really important. And I'm not sure if it's a fair assessment or not, but my impression is that my generation and the younger generations, I'm still, I hope, quite young. I'm 31 now, so I'm not very senior in any way whatsoever. But I feel like people our age are very conscious of workplace culture and things like work-life balance and stuff like that. And I really feel like if you want a lot of people to want to start companies and to succeed, then you have to make doing that sustainable things that people can do and still have a life.
Yeah. People want to feel purpose, don't they?
They want to feel purpose, and they also want to be able to start families and have in some cases more than just work in their life. So I think that's really important to take into consideration.
I think you'll be growing great opportunity for people to join the business.
I hope so, yeah. So right now we are only scientists or people with science training. So it's ourselves, both me and Simone, we have each of our PhD from Jan Terje's group, and then we have one other PhD level researcher, and then one younger graduate level researcher working in the company. So for sure we will be needing to, that's also something I'm looking forward to know how to work with and also manage people with other sets of expertise, other skill sets than myself. That's definitely like project management, and employee management is of course also a big part of starting a company, if you're part of the founding or the executive team.
Absolutely. Absolutely. But it seems like something you're very passionate about. Yeah,
I am. I think that's it's a very important and nice part of my job, I think. Not that I really manage people, so to say, but of course there's certain priorities as chief scientific officer that you ultimately have to make them, or at least speak up and have a scientifically sound opinion. So I think that's cool.
Absolutely. Well, thank you very much. It's been fascinating hearing not just about the work that Authera are doing, but your personal story.
Thank you for the opportunity to share.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium or TikTok. See you next time.